Okay, sure. Okay. Welcome everybody. Thanks for coming for the departmental seminar. And today I'm very, very happy to introduce you Kristin Branson from Janelia farms in Howard Hughes Medical Institute. And let me just start by saying that this is, Kristin is a great example of what happens when a scientist that is expert in a field cross its discipline to try to answer questions from a different discipline. In this case, she's an expert in computer science, and she decided to cross its discipline to answer biology questions. And this has resulted in really great work. So let me tell you a little bit about her. She started actually her major in Harvard University in astrophysics, but then she took some classes in computer sciences, and she liked the problem-solving aspect about computer science, and so she decided to switch her major and then got her AB in computer sciences from Harvard University in 2000. Then she joined Dr. Serge Balonji and Sanjoy Dasgupta in UC San Diego to complete her master's degrees and then her PhD in computer science. And during this time, and even before when she was an undergrad, she spent several internships in NASA. And actually during her PhD, she was a fellow of the PhD program during the whole PhD. And it was during that time that she ended up working in a biology problem. And then she developed a system for automatic tracking multiple lab mice from a side view. That is also when she realized that machine vision and computer sciences could be used to solve biology problems. And then she also saw that there was not many people working on that. And she saw that she could have a great impact in this field. And so that's when she started to working in more directed to biology problems. And then that's when she found her way to work with the best animal model in biology, that's Drosophila. And she joined Dr. Michael Dickinson in Caltech for her PhD. And there she developed a machine vision and learning system to automatically track Drosophila flies walking. After that, this software is called C-Tracks, and it's open source. You can find it online. And then after that, she joined Janelia Farms from the Howard Hughes Medical Institute in Virginia, which is just an hour driving from here. And then she became a group leader. And one of the first things that came out of her lab, this beautiful paper where she developed a new software called JAABA, which I don't really understand a lot of how it works, but I can tell you with my own words what I think this software can do. So this software basically can learn from a researcher intuition. And so imagine you basically have a video recording of an animal behaving. And so the researcher can use its intuition to classify behavior. So the researcher can annotate by selecting frames from this video what his intuition is telling him about a particular behavior. And then the software, what it does is it takes these annotations, these manual labels, and convert them into math and then into automatic classifiers. And then you can actually input large amount of data to the software, and the software will automatically classify the behaviors for you. And so with that, I will let her to tell you more about this and what is she working right now. And thank you, Kristin, again, for accepting this invitation.
Well, thank you for inviting me, and thank you for that really nice introduction, and thank you everyone for attending. So yes, so today I'm going to tell you about some of the work that our lab has been doing on using machine vision and learning to analyze animal behavior. And as Isaac said, I'm a group leader at the Howard Hughes Medical Institute's Janelia Research Campus, which If you don't know it, we're actually very close by. We're in Ashburn, Virginia, so kind of suburbs of DC. We primarily work on neuroscience research right now, although we're in the process of broadening the fields of research that are done at Janelia. And my particular part of Janelia is the computation and theory research area. So we have a group of 9 group leaders who are working in the areas of computation and theory. And our goal is to build quantitative understandings of biology and interpret data at really large scales that are relevant for kind of modern biology. Okay, and so what I'm going to tell you about today is the work that we've been doing on quantitative analysis of behavior. And you can see quantitative analysis of behavior as a tool that's useful in a lot of different biology experiments, in particular neuroscience experiments. So a couple of the types of experiments that we work on, either in our labs or with collaborators, first are experiments where we're able to record the neural activity of the animal. So in this case, we're using either electrophysiology or functional imaging to record the neural activity of a mouse. And we're also able to record video of the animal behaving. And our goal is to be able to understand the relationship between the behaviors that we're monitoring and the neural activity that is generating that behavior. The other type of experiment that we've done a lot of in our lab is using performing neuronal manipulations of the activity. So for instance, we can use things like optogenetics to genetically target specific neurons in the fly brain. And so that's what I'm showing here. In magenta, you see kind of a reference pattern stain of the fly brain, and in green, we're showing the neurons that we're able to target genetically. And we use optogenetics to activate those neurons. So if you look at this video down here, when this light right over here turns on, we are activating those neurons with light. And what we want to be able to do is look at the changes in behavior that we see when we activate these neurons. So if you are used to looking at flies walking around, you can maybe tell that all these flies start walking backwards when we activate these neurons, and that can help us understand that these particular neurons are involved in the control of backward walking in flies. So quantitative analysis of behavior is a useful tool in neuroscience, as well as lots of fields of biology, anywhere where you want to take quantitative measurements of animals behaving. But I actually am kind of interested in it for its own purpose. I think understanding animal behavior is one of the fundamental goals in biology. And as a computer scientist, one of the things I think that really interests me is that these simple organisms like flies are able to do really complex behaviors, things that are more complex than what kind of the most complicated neural network trained by people at Google or DeepMind can do, with really minuscule brains and really small amounts of energy used. So they can navigate their environment, they can find food and mates, they can avoid predation and survive in the wild, which is not anything that an artificial algorithm can do right now. So we'd really love to understand the algorithms that animals are using to survive in their world, and why those algorithms are implemented the way that they are. So some of the things that we try to answer in our work are, what is the vocabulary of behavior? Can we come up with a set of words and structure to describe behavior? And what are the rules of behavior? Can we come up with some rules of when an animal sees this, or when it experiences that, it performs this kind of behavior? So that's the type of thing that we're looking for in our research. And as I mentioned earlier, we use video to collect information about the behavior of animals. Video is great in a lot of respects. It's non-invasive, inexpensive, and easy to collect. And for us as humans, it's quite easy to interpret. We can see these videos and we can kind of tell what's going on. But to actually get a quantitative sense of this data and to convert this into meaningful numbers, we actually have to think of this video. This is an image that's 1,000 by 1,000 dimensions. So it's in the million-dimensional time series of data that we're looking at. Being able to quantitatively interpret this is actually quite a hard problem. That's the focus of computer vision. So our goal is to be able to take video like this and convert it into some kind of quantitative, low-dimensional, meaningful, and interpretable description of behavior. And so the questions that we try to answer are, what should this representation be? And once we have a representation that we like, how do we automate its computation from video? Okay, so to answer this question of how should we represent animal behavior, we first, let me just start by saying we don't know. We're still working on this problem. But some of the things that we're thinking about are one dichotomy is whether we want to use a discrete representation or a continuous representation of behavior. So in a discrete representation, we might say that there are 3 different things that a fly can do. It can walk, stop, and turn, and it's doing one of those things at all times. If we, on the other hand, we could choose a continuous representation. And here, instead of having just 3 different things that a fly can do, we have maybe 2 properties that we think are important, but they take real values over time. So we might say that the speed that an animal is moving at and the distance to another animal are the 2 properties that matter to describe behavior. And so that's one continuous representation of behavior that we could choose. If we choose a discrete representation of behavior, where do we get these different categories? How do we come up with what the categories should be? So one thing that we've relied upon is, as Isaac was mentioning, is biologist intuition. We can ask biologists what the behaviors that they've seen are and have them define some behavior categories. And then we can use supervised machine learning to automate the computation of those categories. Alternatively, we can use unsupervised learning to try to learn what the behavior categories are. So let's say that we've decided that the 2 properties that are important for describing the behavior of flies are their speed and the distances between animals. We can do things like clustering to be able to come up with groups of behavior categories. For instance, k-means clustering allows us to come up with this clustering of behaviors into categories. And when we say that speed and interanimal distance are the 2 properties of behavior that matter, what we're doing is we're mathematically defining what it means for something to be a good representation of behavior. So in both approaches, we have to make assumptions. We have to impose those assumptions. In supervised learning, we have these explicit assumptions that walking, stopping, and turning, say, are the behavior categories that flies perform. Versus in unsupervised learning, we're saying speed and interanimal distance are the properties about behavior that matter. And these is exactly how we are going to weight those different properties when we perform a clustering. So there's no right answer about what the right way to do this is, and we're kind of exploring both of these different types of approaches. And I'll try to tell you about a little bit about both of the ways that we're doing this. So in the first type of approach, the supervised machine learning approach, we're trying to do things that are relatively easy for a human to do at kind of small scale. So for instance, if we can watch this video of a mouse eating a food pellet, maybe we can relatively easily say, it's lifting its paw, it's grabbing a food pellet at any time point, or vice versa. We can say that, exactly where the feet of these flies are, of this fly is, but it's hard for us to do that at the scale that we want to do biology experiments. So we have some experiments that I'll talk about where we collected 20,000 videos of flies walking around, and that's too much data to be able to do that kind of analysis manually. So our goal in these supervised techniques is to be able to automate things that are relatively easy for a human at scale. And we focus really on developing usable tools for biologists here, and I'll try to emphasize that when I tell you about these approaches. The other type of project that I'd like to talk about is unsupervised machine learning, where we're trying to discover structure. So we want to do things that maybe are hard for a human to do, things that a human maybe can't do, find new structure in data that we hadn't hypothesized as existing before. And I'll tell you about some very early work on doing that, where I have no actual results, but just kind of I'll tell you about the approach that we're trying. Okay, so I thought I'd start telling you about what Isaac was mentioning in his introduction about behavior categorization. So here, the goal is to be able to assign for each fly what it is doing in each frame. So in this case, we've come up with 15 different behaviors that flies can perform. And for this fly that's annotated with a triangle, we're showing the behavior that it's classified as performing at every time point. So we developed some software called JAABA to be able to train machine learning classifiers to automate behavior categorization. So I'm going to show a screencast of me training a wing grooming classifier. So for those who haven't looked at flies behaving, wing grooming kind of looks like you can see kind of the wings bending underneath the fly. That's kind of what you can see at this resolution of video. And so what I'm going to do is I'm going to label a small set of frames where the fly is grooming its wings. So this timeline here represents the manual annotations from the expert biologist. So I'm labeling that the fly is grooming its wings here, and then I'm going to label that it's not grooming its wings in another time period. And you'll notice that I left this little gap here because it's quite hard actually to say where a behavior starts and ends. It's kind of hard to say exactly when the fly stops grooming its wings. We've also tried to make the machine learning relatively fast, so you can train a classifier in about 30 seconds to a minute. And what this down here is indicating that it is training the classifier right now. And then the predictions of the classifier are being shown in this timeline here. And so we made this software interactive where the user can kind of scroll around and look at the behavior to find examples where the system is predicting the behavior incorrectly and the biologist is sure of what the behavior is. So from just those kind of two bouts of labels, maybe the system has learned that to be grooming, a fly needs to be sitting still, but it hasn't quite learned that its wings need to be moving in a specific way. So I can correct the annotation, or sorry, I can add new labels where the system is predicting incorrectly. And then I can retrain a classifier again in a few seconds while I wait, and then look at the predictions again. And so we've spent a lot of time kind of making this GUI really easy to use. And so as we developed methods to categorize these behaviors, we learned a few lessons that guided our development. So as I said, we wanted to make this software something that biologists could use to train their behavior classifiers. And the reason for that is that we don't know what all of the behavior categories are. We can come up with a list of 15 behaviors in our lab that we think are relevant, but the behavior definitions might vary by biologist. You know, there might be different conditions that you see different behaviors in. Behaviors might look different in different environments. So we wanted to make this something that biologists could use on their own to train behavior categories. Another thing that we've done that seems maybe very intuitive, but was something that nobody had actually done before, is we didn't require biologists to label every single frame of the video. So what people had done before is just say, okay, here's a video, label every behavior and every frame that you see. And there are a couple reasons why this is an inefficient way of doing things. So the first is that what's called a long-tailed distribution in machine learning. So some behaviors like walking and stopping are very common, but some behaviors like jumping or chasing might happen a relatively small fraction of the time. And if you want to get enough labels of these rare categories where you're labeling every frame in the video, you're going to be labeling a lot of data. So in the past was actually quite difficult to train classifiers for these rare behaviors just because we didn't have enough annotations of them. So we made it so that biologists could just choose which frames they label, and we made it easy to scroll around in a video and find different flies or different videos or different frames to label to find enough of these examples of rare behaviors. Another thing that we found is that biologists are often unsure of what the right label is in some cases, and that might be because their definitions of the behavior are not quite as crystal clear as they originally think they are, or it might be because the resolution of the video is limited. But what we found is that biologists would spend all of their time trying to annotate these difficult to annotate frames and not do it in a consistent way either. So we just said, don't label those frames, let the machine learning do something reasonable on it, and that hope that everything is okay.
Hey, Kristin, can I interrupt you?
Yes.
There's a couple of questions in the chat, and I think they're good for stopping now. So Heidi is asking if will the gap between the coded behaviors make it difficult to understand the underlying changes in the neuron signaling at this critical moment?
It's a good question. I think I'm not going to show you numbers about the accuracy here. I would say that the accuracy of the classifiers is pretty close to the accuracy of humans. It's not quite as good, but pretty close. And the hope is that we have enough data that those misclassifications are going to kind of average out and not have a big effect. What we end up doing is computing statistics over large amounts of time and over populations of animals. And if there aren't biases in your classification system, things should become pretty accurate as you average more and more data together. And we've looked for biases in our data, and that is something that there is something to worry about. In particular, when one of the things that I'll tell you a little bit about is that we wanted to use these classifiers on the behavior of different genotypes of flies, where we're activating different neurons. And we might see a behavior that we've never seen before in the test data that we haven't seen in the training data. And then the machine learning can do quite weird things and return biased results. And so we had to be very careful of that when we used the software. It's important to review how well things are actually working.
Okay, there's another question, and I just remembered that you told me that you wanted to have a more interactive seminar. So I'm gonna ask.
Sure, please.
Just yes. Speak up about. I don't know if I'm pronouncing your name correctly, but can you...
That's correct. So I'm curious if the software can be used in non-laboratory settings. So, for example, drone footage of animals in the wild.
Yeah. So the way that this particular version of the software worked that I'm presenting is it works from trajectories. So you have to have first tracked the animals, and then you put those trajectories into the behavior classifier. So any type of video that you can track the animals in, then we can do this kind of behavior classification, and that should be fine. But I've kind of hidden the problem in the tracking problem, right? And tracking becomes harder the more variability there is in kind of the environment that you want to apply this to. So there's a lot of work that people are doing, and we're also working on to try to, get tracking to work better. And one of the things that I'm quite interested in is how we kind of can get out of the lab. These software are working pretty well in the lab. How well will they work outside of the lab?
And Heidi, you have a following up question about the gap. You wanna?
Yeah, I'll just unmute. Kristin, thanks for your answer. My question about the gap was, you were saying that in that fly video, you were intentionally like stopping coding one behavior, and then there was like a small moment in time because it was hard to tell when one behavior was ending, the other one was beginning. But it seems like at some level, that moment of transition between behaviors would actually be like the super cool part, but that you're kind of leaving as a blank.
Yeah, no, that's a good question. And actually that's something that we've been working on more recently is, so here we're kind of saying every frame, is the fly doing this behavior? Is it doing that behavior? And the things that we were generally looking for is the fraction of time that the fly spent doing behavior X. So in that case, those kind of small frames in between don't matter because we weren't looking at the temporal patterns of them. But more recent work, we actually care about, as you said, kind of associating changes in the behavior that's occurring to the neural activity that we're observing. And so what we've started doing is, instead of trying to do per frame classification of what the fly is doing at each frame, we're trying to do classification of the start of new behaviors. So then, you don't need to necessarily get exactly which frame it happens on correct. You just need to know that a behavior started here. And you want to get it as correct as you can, but we don't have a perfect definition of the behaviors. And that's kind of this chicken and egg problem of like, can we maybe use the neural activity to get a better sense of when decisions about changes in behavior are made, or when behavior is actually changing versus the other way around.
Okay, thank you, Kristin.
Sure. Okay, so yes, we use this this relatively simple kind of labeling that seems to make sense, but it actually hadn't been done in computer vision to my knowledge before. And so this is a kind of active learning in machine learning. And then the final thing that I mentioned in response to Heidi's question, but I think was not exactly what she was asking about, we do this interactive training. So the kind of basic assumption in machine learning is that the data that you label, your training data set, is a good representation of the data you actually want to apply your learned classifier to. But that's of course not the case in any kind of real life application, but particularly in biology. Let's say that you only train on flies in one condition, and then you come up with some other condition that you want to apply your classifier to. There's no guarantee if your data set distributions are different that that machine learning classifier is going to work particularly well. And that's the case for us, where we have thousands of different genotypes of flies that we want these classifiers to work on with quite diverse behavior. So our data are very non-homogeneous. So the example for that is some work that we did with the original Janelia Gal4 collection, which is a collection of genotypes of flies that allows us to genetically target sparse subsets of neurons in the fly's brain. And activating these neurons with thermogenetics results in quite different behavioral effects. So these are just a few different examples. So in this Gal4 line, we see a lot of courtship between flies. In this Gal4 line, we see a lot of jumping between flies. In this Gal4 line, we see a lot of aggression between female flies. And if we only trained on normal flies, control flies, we would see very few cases of these behaviors. And actually, we never see female aggression in our control flies. So if we only trained on kind of normal flies, our behavior classifiers would not generalize particularly well to these different types of genotypes. So we actually came up with a way of interactively building our training data set, reviewing the results of the behavior classifiers and repeating. And so that allowed us to apply this to the 2200 Gal4 lines that we had collected video for, which was 20,000 videos, almost 500,000 flies, and 10 billion fly frames. So we've been taking a similar approach to create something called the animal part tracker, with the goal of being able to track parts of flies, or parts of animals, I should say. So the idea with pose tracking is you have an image of a fly and you want to automatically identify where a set of landmark points are. So for instance, we might have said that for these flies here, there are 17 points that we think are important to identify in a fly, and we want to know where those points are in the video. So as I said, there are kind of two components to our software that we think a lot about. One is, of course, the machine learning part of this software, kind of what algorithms we use to train these classifiers. But I think what makes our lab quite unique is we spend a lot of time thinking about the user interface of how you actually get enough good labeled training data to do a good job training these algorithms. So kind of the labeled training data and the algorithm are the most important things for creating a successful classifier. And to get good labeled training data, you need a good user interface. So this is a screencast of Alice Roby in my lab training a part tracker for walking flies. And so she's going to label the 17 points that she's identified as useful to track in a fly. And we've made the software, again, something where you can switch between different videos. You can switch between different flies to label. So what it's showing right now is switching to a different video and switching to a different fly to label. You can also, so there are a lot of hyperparameters involved in machine learning, and some of them actually matter for getting your machine learning to work. And so to try to make this something usable by biologists, we've made visualizations of the effects of those different parameters on, for instance, here, data augmentation. The other thing that we've done with this software is we've tried to make it easy to both train and visualize the trained classifiers. So you can hit this train button and it will connect to a GPU to be able to train a tracker. You can look at these trackers are trained with an iterative algorithm that's kind of decreasing some criterion of how well things are doing over time, and you can kind of look at that. You can hit the track button while it's training to look at intermediate versions of this tracker. That's what it's going to show next. So yeah, you can hit the track button. And so again, the goal is to have this kind of interactive framework that allows us to get useful data to label, as well as to be able to let the biologists know how well things are actually working. So just maybe one slide about the algorithm that we've been developing for doing this, this tracking. The most successful algorithms in computer vision right now are based on deep neural networks, deep convolutional networks. And so that's what our algorithm is also based on. And the way that our algorithm works is it takes an image of some flies. And the thing that's unique about it is it tries to produce a bunch of different hypotheses of where the flies are. And it does that using relatively local information. So at this particular grid location, it's going to look around that grid location and try to predict where the parts of a fly are. And it's going to do that at every single one of these different grid locations, and we get a different hypothesis of where the flies are for each of these grid locations. And then we also have another output, which is which of these grid locations should have a lot of weight. You know, how confident are we in each of these grid locations? So we do a combination of kind of taking the different, weighting the different hypotheses and using those. So that's kind of the way that our algorithm is working. But there are a bunch of different algorithms for pose tracking. Pose tracking is quite a popular problem in both biology applications as well as in computer vision. And so we wanted to be pretty forward thinking on that front. We want to acknowledge that there are a lot of different algorithms that are being developed, and we're just one of the labs developing those algorithms. So we've tried to make our software something where you can kind of plug in and implement a bunch of different machine learning algorithms for predicting the pose of the animal. So one that you might have heard of is Deep Lab Cut. That's kind of pretty popular in animal tracking, and Open Pose is quite popular in the computer vision literature for human tracking. So we have implementations of both of those built into our system. So to make this something that's easily extendable, what we did is we kind of tried to take the parts of our system that were very specific to our system and our type of data. In particular, those are things associated with grabbing images from video and something called data augmentation, where it's a way of kind of automatically increasing the size of your training data. So this stuff is something that's quite particular to the type of data that we work with. But then there's a part that's very general to any kind of pose tracking problem, whether that's human pose tracking, animal pose tracking, anything. And so that part of the code is something that we've kind of abstracted away everything that's very specific to our problem and made it something that can fit into other people's code quite easily, so that we can adapt new algorithms into our software easily as progress is made. The other thing that we tried to do to make this really usable is think about how biologists would actually be running this software. So deep learning algorithms all require pretty high-end compute architecture. So you need expensive GPUs basically to be able to run this, and potentially a lot of expensive GPUs to be able to run these types of things at scale. And that needs to be integrated on different operating systems. And some people have a lot of GPUs locally. What kind of more common solution is at kind of bigger institutes is to either have a GPU cluster locally at your institute, or to use cloud computing like the Amazon cloud or the Google cloud. And so we've integrated being able to connect to all of these different types of hardware into this software that we've been developing. So you choose an algorithm, you choose a backend to run that algorithm on, and so the requirements of what types of hardware you need to have in your lab are relaxed. So we've been doing a lot of ground truthing of how well these different algorithms work on lots of different types of applications. We have a few different types of applications that we work on, either in our lab or in collaboration with others. And so those include tracking multiple animals from a single view, or tracking one animal from multiple views with the goal of being able to track in 3D. And so some of the types of animals we've worked on are flies, mice, and larvae, which are the main types of animals that we work on at Janelia. To kind of look at how well this is working in practice, to kind of see, you know, do different algorithms perform differently? Have we solved the problem? We've done a lot of ground truthing where we've manually annotated the correct answers on some test data that's not part of our training data set. And one thing that's quite hard is actually to say what it means to be correct in your estimate of a joint location. Like how far off can the, how far off should the error be to be considered correct? So what this plot is showing is for different thresholds of what the threshold of what is considered correct or incorrect, how well different algorithms are doing. So what the accuracy of these different algorithms are. So just to shorten that, the higher the value you have, the better. And it's kind of a question about where exactly along this x-axis you want to be. We probably want to be, you know, somewhere in this cyan box that I'm showing right here. So this is 5-pixel error, and that's what a 5-pixel error looks like on a fly in our data set. So we kind of want to be somewhere in this box, and actually in this box we see a little bit more variability in how well different algorithms work. And this MDN algorithm is the one that I told you about. The gray and the black lines are how well an annotator can annotate the same data twice, and we compare kind of intra-annotator and inter-annotator error. So humans are still better at this task than the system is, than the automatic system. You know, whether this is a meaningful difference or not it kind of depends on the application. I'll skip through this. This is just kind of for a bunch of different data sets, how these different algorithms compare. And yeah, just to kind of end here, as I was saying, you know, the human annotators do a better job than all of these algorithms do, no matter kind of how much training data we've tried to give these algorithms. So this is if we only train on 5 training examples versus if we train on almost 5,000 training examples. And we're still well below the accuracy of human annotators. So, you know, is the problem solved yet? And I think the place where things are really not working particularly well is when you have occlusions or you have interactions between different animals. So this is an example here of a tracker not working particularly well. So from this particular frame, you know, you've got all these fly legs all over the place, and it's kind of hard to tell which leg is which and whose legs belong to whom in this particular example. And actually for a human annotator to do this, what they actually do, not play. Okay, so this is, let's see if okay, so this is a video. It's be able to play? Okay, there we go. So if you look at kind of enough temporal context of what's happening here, and maybe this is hard if you're not used to looking at flies in video. I've been looking at flies in video for the last 15 years. You know, when you have enough temporal context, it actually becomes somewhat clear what's going on, you know, which leg is which. And that's something that's not particularly well exploited in computer vision algorithms right now. Almost all of them operate on single frames independently. And so we can do a lot better if we start taking into account that this is actually video. We have more information than we're using. So that's one of the research directions that we're working on. Okay, so I'm going to switch gears here now. Are there any questions before I do that?
Hey, Kristin, I had a couple questions. Thank you for this, by the way. Appreciate you joining us. I was curious, first of all, how does your software account for movements in the z-axis as opposed to XY, which is, it seems like what we're looking at mostly? Yeah,
so right now, the only way we can get XYZ locations is actually to have multiple views of the animal. So we'll have multiple cameras that are set up, and then we do triangulation to figure out the positions. Otherwise, everything is done in 2D. You know, the data that we collect in our lab is flies on a plane. So, you know, assuming, you know, yes, there's a little bit of movement in Z, but quite a small amount. But it actually is something that we're interested in working on. We have a collaboration with DeepMind right now where they've been building with us a 3D model of a fly. And so we're hopeful that we might be able to pull out, 3D information from a 2D view by having an accurate model of a fly. Or we just need to really build a fancy setup with a lot of cameras.
Right, right. And so then my other question was about other qualities or other behavioral outputs aside from electrophysiological or fMRI data. And I work in the auditory system, so I was thinking about sound output and whether something like that could be paired with video footage. I'm not sure how noisy flies are, but maybe they make some shushing of the wings or some noises or something.
So there are people who work on fly songs. So there's a courtship song that flies perform, and so people try to look at that with flies. We actually work on with mice. We are looking at ultrasonic vocalizations and the movement of mice, and we're trying to understand the relationship between those two. That's one of the biologists, Roy and Egnor, in our lab is working on that.
Cool, thank you.
Any multimodal signal is super interesting to us because we're hopeful that kind of one signal can inform us about the other.
Right.
Kristin, I have a related question. Similar to what Tom was touching upon, when you go out in nature, you have this soundscape which can be incredibly complex. You might have 50 bird species singing at once. And so you've got these complex layers that are really hard to differentiate in a sonogram or a spectrogram. And I'm wondering if you know much about using machine learning to sort of help differentiate the soundscape to identify species, whether it's birds or bats or flies.
So I've never worked on this. What Royan has done is the kind of engineering approach, and that was because she wanted to know which of her mice was vocalizing. So you didn't have different species that make different sounds. Potentially, they all might sound the same. We weren't totally sure to begin with. So she just had lots of microphones and was able to try to, there's a step of kind of segmenting, this is a vocalization that's happening, and I'm pretty sure it's only one animal producing this vocalization, and then assigning the identity there. I think the question you're talking about is much more complex, where you have lots of animals making sound at the same time. Yeah, exactly. I don't know of anyone who's worked on that myself, actually. And yeah, that would be interesting to work on.
Yeah, and just to keep thinking in the future, to integrate the visual and the auditory to collectively to automate just identifying things out in nature would be amazing.
I was just going to say there is some vocal classification of using machine learning of marine mammals, but it's not like a 50 different species singing, but it's like, we've got these recordings and we're the Navy and we want to blast sounds out into the ocean. We need to know what species are here.
So I know that, so my PhD and postdoc advisors work on the Merlin bird app. It's identifying species of birds. And so they've recently added a feature where you can upload sound of a bird call and try to identify based on that. And they're trying to give in spurters to like get these microphones and go out into the world. But yeah, it's something that I don't think there's been a lot of work on. And actually that's quite interesting to think about kind of how, the soundscape will actually affect the behavior of the animals around you too. I'm sure it has an effect.
Yeah, thank you.
Hyen, do you have a question?
Yes, thank you. Hi, Kristin. I'm actually wondering, like, I know your software right now uses mostly mouse Drosophila, and I'm wondering if it would be possible or easy to adapt it to a different animal. We work with tadpoles, and I'm really interested in trying to characterize basic behaviors, like exactly like what you were describing at the beginning of the lecture. But I'm just wondering if you have anything in your pipeline that could potentially be useful for us.
So we've, we're somewhat limited being at Janelia because the kind of the biggest animal at Janelia is a rat, and there are no tadpoles here currently. There are zebrafish, but we haven't actually worked on them. But we try to make our software as general purpose as we can. There's always, some work in getting it to work on a new setup, but it's something that we would we endeavor to make our software general purpose. We have like 3 different types of animals, flies, larvae, and mice that we test things on, and we hope it will work on another system. But I'm very happy to talk with you about how to adapt it to your data.
Sounds great. Thank you.
Tamara, do you have a question?
Yeah, hi. I was wondering if the program is able to compare and analyze unlabeled frames. So would it find similarities between frames that you were not assigned to any behavior?
Yeah, so we've looked a little bit at that kind of, whether some of the features that are learned as part of training this classifier could be useful as a general representation of behavior. The original way that we were doing this machine learning, so this is a project that we started in 2010, which is pre-deep learning revolution. So it was using something that maybe wasn't as well suited to learning new representations. It's using an algorithm called boosting. But that's kind of one of the goals in our new work, is to try to learn good representations of behavior that maybe you kind of learn this manifold of what behavior that behavior lives on, and are able to say these are 2 similar behaviors because I've kind of encoded some amount of information from the biologist, and I'm trying to have some amount of information learned from the data. It's something where we're very interested in. We haven't been successful on it yet, I would say, but it's kind of one of the main goals in the lab.
Thank you.
Mark?
Yeah, hi, Kristin. That's really interesting stuff. So the thing about encoding the z-axis information is interesting, because the people who do super resolution, one of the approaches to do that is to use a cylindrical astomatic lens in the light path.
Okay.
So that things that are above the focal plane get stretched in one direction. If they're below the focal plane, they get stretched in the other direction, orthogonally, to an amount that basically encodes where it is in the z-plane, in the axis. And I'm just wondering if you could put that in here, since at least for the tips of fly legs, right? Those are pretty discrete objects. And so you could maybe get the 3D information from that kind of an approach.
Okay, that's a good thing to consider. And we talked to some of the people who use like structured light to, there's some commercial systems that use structured light to try to get 3D information. They were trying them on some dead flies to see if it worked, and they didn't give us any particularly useful results. So we weren't confident in it. One thing do you think it would work with kind of relatively fast temporal resolution? Is there any kind of temporal constraints?
No, I think you get it from, you don't have to take a stack.
A single
It's
image?
not structured light.
Okay.
So it's just, since you have a pretty good model for what the fly looks like, you know that if the tip is spread out in one direction, that's a measurable amount.
Okay.
If it's spread out in the other direction, that's another measurable amount. And so that gives you its position in Z.
I will ask so my partner is actually a microscopist, so I will ask him about, he's never mentioned it to me as a possibility. He does light sheets, so maybe it's not his thing, but I will ask him about it to see if that's an approach that we can try. Because
well, you're in the right place, right? Just
Yeah, exactly. But no, I mean, it's like we're thinking about how should we build this next rig to kind of get 3D information in a relatively large area, and there's a brute force, like put 80 cameras on your system and collect a lot of data. But if there's something smarter, I would love to learn about it. Thank you.
I think that's it for now.
So I'll just breeze through this last bit pretty quickly. So what I'm going to tell you about right here is trying to use this unsupervised machine learning, and some of the questions were kind of alluding to this. Can we, not just repeat a human, but can we actually learn, what it means for 2 behaviors to be similar and do clustering, things like that. And so the kind of standard ways that people have used unsupervised learning on behavior analysis in the past are clustering and dimensionality reduction. And so both of these approaches rely on you having some notion of what it means for 2 behaviors to be similar. You have to be able to measure how similar 2 behaviors are. And that matters for clustering when you're saying that these belong to the same category, or it matters for dimensionality reduction when you're saying that this this is a good reconstruction of my original data from a low dimensional representation. So you need this representation to be known already. And so the way that people have done this in the past is they've carefully engineered clever representations of behavior data and then done clustering or dimensionality reduction. And that's done interesting things. There's a lot of interesting work on that. But that's not been kind of what the deep learning revolution in machine learning has been about. That was kind of the standard approach that people took for supervised machine learning when I first started doing machine learning back in the early 2000s, which is you'd engineer a complex set of features that you thought would work well for this particular task, and then you'd train a very simple classifier to be able to do your task. And the thing that's been important with deep learning is that it's deep, that we have these intermediate representations that are being learned as part of the process of your supervised learning approach. And so we start with a raw representation of the data. We start with the pixel image, and we learn a very complex representation of the data, or a complex function of the data that involves more and more refined representations of the information in there. So I'm interested in whether we can use approaches like this to learn good representations of behavior. So we've been exploring something called behavior forecasting, which is a kind of in the realm of self-supervised learning, if you are into the machine learning keywords. And the goal here is to be able to take some information about what a particular fly, what it's seeing, what it's sensing, and be able to predict what it's going to do in the future. And the idea is that if we have a good model of predicting what a fly is going to do in the future, then we've learned something quite important about behavior. So just skip through this. So here's the type of data that we've been applying this to is video of 20 flies, so 10 male flies and 10 female flies walking around in a dish. And we've tracked these flies in the past using the C-Track software that I developed when I was a postdoc. And our goal is to be able to train something that can predict the future trajectories of flies from its past trajectories. So why would we want to do this? Okay, so if we learn a really good model that perfectly predicts what an animal is going to do, and that machine learning model works the same way that the animal does, then just understanding this machine learning model means that we've understood animal behavior. So that's kind of pretty ideal. There's a couple of questions there. Does the machine learning model work like the animal? And another big unknown question, it's very hard to actually understand what these deep neural networks are doing. So we need to come up with ways of interpreting this machine learning model. So these are kind of the two things that we've been working on is, creating complex models that do a good job of predicting the future behavior of a fly, trying to figure out how we make sure that those machine learning models work like an animal, and then how to, thinking about how we do this interpretation of complex machine learning models. So I'll skip through kind of the details of exactly how we're doing this. I'll just say that we're training recurrent neural networks to do this. And the idea with a recurrent neural network is that you learn a state that describes everything that's relevant about the past of an animal in predicting its future. And one of our hopes is that that state that's being learned is a good representation of fly behavior and something that we can analyze in the future. Okay, so we've spent a lot of our time just thinking about this first question of, does the machine learning model work like a fly? And there actually, hasn't been, I think that's one of the, primary places that we need to really be working in unsupervised learning is really telling whether we're doing a good job or not with our model. So a couple of the ways that we've been doing this, so we can, use this machine learning model that we're training to predict what the fly will do in the future, but we can actually do that a bunch of times. So we can predict what it will do one frame into the future. We can feed that back into our system, do another prediction pretending that the fly had moved there, and continue. And that allows us to estimate how well we are predicting the behavior many frames into the future. And just to kind of show you a simulation of that. So the blue flies are all real flies. The blue triangles are all real flies. And this red fly here is a simulated fly that we've made with our recurrent neural network. So just to I know most of you are not experts on fly behavior, but these are 4 different videos. 3 of them are simulations, and 1 of them is a real video. And the red fly is the only one that might be simulated. So can people tell which of these videos is the real video? Okay, so the real video is this one in the bottom left here. And I've been looking at flies walking around in a dish that looks a lot like this for the past 14 years now. And it's quite hard actually for me to be able to tell the convolutional net and the recurrent neural networks from the real fly. So we've done a lot of quantification of how well these things are working. We're getting to the point where things are working pretty well. We think that there's a little bit more work that we can do here to kind of make the systems work better. So one of the things we're doing is kind of trying to make the systems work better. But the other thing, and I'll skip through this just in the interest of time, that we are spending a lot of our effort on is this kind of the second question that I posed. So if our system works like a fly, how do we learn something from that system? Did we just train a really complicated deep network that is even harder to interpret than a fly, and now we've gone down this other direction that is not going to be helpful? Or can we learn something from this model? And so there's this whole field of interpretable machine learning or explainable AI that we're working on tapping into to try to understand this. So I'll just end by putting a plug in here for Janelia. So Janelia is a fully internally funded biology research institute with a focus on new imaging tools and techniques for analyzing and understanding the resulting new types and scales of data. And we have this computation and theory research area that I'm the head of, and we are always looking for new lab members. In particular, we have a program called the Janelia Theory Fellow Program, which is an independent postdoc position where you can kind of work with multiple different labs, both on the computation and theory side, as well as experimental labs. And I'll just end there with thanking all of the people who did this work and my collaborators.
Thank you, Kristin. Thank you.
Thank you.
We will start with questions now. Finn, do you have a question?
Yeah, thank you so much, Professor Cervantes, and thank you so much for the wonderful presentation, Dr. Branson. So, as a biology and comp sci double major, I am really interested in how various computer science methodologies can be used to collect, analyze, and interpret data. And personally, I plan on using comp sci to enhance my studies of genetics in the future. So, in my undergraduate comp sci classes, we've discussed in depth the difficulties of space and time trade-offs and how many algorithms cannot be as thorough or effective as we'd like them to be due to hardware limitations and processing times. My question is, in developing and utilizing this machine learning software, what limitations have you experienced with the software, and how do you think that further advancement in computing power and hardware could allow for more accurate tracking and modeling?
Yeah, I think it's a great question. So we've kind of been in the approach of just kind of throwing more hardware and more time at our problems right now. So there's neural networks are very slow to train, right? Like some of these things can take weeks, months to train the neural network. And right now we haven't been spending a lot of effort ourselves on trying to make that faster. We're kind of relying on other researchers to do that, and people are working on that, right? I mean, the big step that actually led to this deep learning revolution was implementing deep learning on a GPU, right? It's just too slow to do on CPUs, but if you put it on GPUs, then it's fast enough. But we are quite limited actually in the complexity of the models that we can train with these kinds of systems. And one of the things that I'm actually pretty concerned about is that there's this kind of hardware divide, right? So like Google and DeepMind, have, I don't know how much GPUs that they throw at these problems. I mean, I think there was an estimate that training the original AlphaGo cost $10 million in compute time. And that's not something that, an individual lab is going to be able to do, right? So it's only a few places that can actually implement that kind of an approach. One of the things that I touched on in the work that we're doing that we are quite limited on is these kind of recurrent neural networks and doing things with video. So most of the computer vision approaches that have really been working well are things that work on individual images, because you can kind of fit a bunch of individual images into the memory of your GPU. If you start going to video, video doesn't fit particularly well on a GPU, and we are very limited in the amount of temporal context that we can incorporate because of the way that networks are trained. And coming up with smarter, whether that's coming up with smarter algorithms or better hardware, I don't know what the solution is. We're not a hardware lab, so we just kind of use existing hardware and we try to come up with better algorithms. But it's definitely one of the things that is a headache for us right now.
Janet?
Yeah, you mentioned, wanting to bring some of this technology in and machine learning approaches into the field. What are the kinds of problems or topics that you think would be amenable to this type of work?
Yeah, I'm definitely not an expert on that, and I would love to hear your opinions on that as well. I mean, I think, given enough training data, we can probably track, we can kind of do all of these supervised approaches in the real world. I think the thing I hadn't thought about is the soundscape of lots of different sounds. I definitely haven't thought about that, but I think we should be able to, track things that anything a human can track in the real world. I would hope that given enough data, we would be able to kind of do that as well. I mean, there's a lot of engineering, I think, that's involved in getting that to happen. But I'm really open to ideas of, ways to find a compromise between what will be relatively easy to implement in the natural real world and what is actually interesting to do. And I'm not an ecologist. I don't have expertise there, so I would love to learn.
Well, the sound would be more amenable because you can, you have a recording and can get you can kind of get the whole pattern within a certain range, but with visual data.
Yeah, so people do like camera traps a lot. So you can look at you don't get information about everywhere that these animals are going, but you get a lot of information about a small area, and you can hope that interesting stuff will eventually happen in that small area that you're looking at.
Yeah, I mean, one potential is like the impact of different types of sort of sound pollution or human-made sound pollution, how they impact different species, like in their acoustic behavior, would be, I think, because usually, like with recordings, you have like such a tremendous volume of data, and like a human can't go through it.
Yeah, that would be an interesting thing to think about. Yeah, like I said, these are things I'm like interested in, but I've learned biology. I've not actually taken a biology class. I've only learned it by interacting with biologists, and so I've learned fly neuroscience, I think, pretty well. But the rest of biology, I would love to learn from other people.
Leslie, do you have a question?
Thank you so much. That was just fascinating. I had a question about, and actually one thing before I ask this question is following up on what Janet was saying in terms of bringing stuff into the field. There's tons of tracking data out there where they put like GPS units on different organisms and follow their movements around, and you can kind of imagine trying to use all the environmental data out there to try to figure out what it is that they're actually responding to. And in fact, there's this whole class of models, individual-based models or agent-based models of trying to figure out how organisms are moving around in their landscape. And so I think those are all areas that are really ripe for that kind of analysis. What I was wondering, I always wonder about these machine learning algorithms and whether or not, the underlying quote unquote results are something that make sense to us or are meaningful to us. So for instance, after all of this work, training these computers to understand fly behavior, is there something that you can articulate, like as a fly moves around a dish, what is it responding to? Is it like, I want to get as close to a female as possible if I'm a male, but when I get this far away, I'm going to turn to the left? I mean, is there some kind of way that you can explain to us what flies are doing?
So we don't have those results yet. I think, like, you know, one thing that's quite hard is that biologists are very good at this, right? And biologists have been looking at fly behavior for quite a while. My advisor in my postdoc, Michael Dickinson, you know, would just stare at these flies walking around and be like, oh yes, the fly is using this algorithm to into some other fly. And I'm like, I don't know how you got that, but I believe you. And then we would test it out and it would be true. And so, you know, there is a lot of expertise that people have. And so what we need to be able to do is beat, you know, experts at this to be useful, right? Or we need to have different biases that we're bringing into the exploration. And so our goal is to be able to do that. We don't know whether it will be possible or not. And it's that's kind of the risk with this approach, is we don't know whether it will work.
Is it okay that I root for the experts always being a little bit better than your machines?
You can root for it. I think you might be better. We might have different biases at the very end.
Thank you.
Pete?
Leslie stole my question, but I maybe just build on it a little bit. I just feel like at this point in time where the haystack is getting bigger and bigger and the needle is staying the same size, the more we can use things like machine learning to continue to interpret things like the natural world, and at the same time make these complex systems really accessible to more and more people. I think it's really exciting, and I think it's really fun to think about how we can use this to do that, to scale up what we know about complex behaviors and make it more accessible to more people. So I think this is really exciting, and it's on the edge of this interdisciplinarity, which is just phenomenal. So thank you very much for your talk. It was really wonderful.
Yeah, I mean, I think that brings up a point that, like, I've tried to do with our work, is to acknowledge the expertise that humans have and to try to figure out the best way of combining human expertise with what machines are good at. And machines are good at doing a lot of things a lot of times and having a good memory, and, you know, having infinite patience. So, you know, how do we combine those types of things to look at new types of data?
Yeah, and given that my memory is getting worse and worse every day, I'm clinging to this to this expertise. Thanks, Kristin.
We have another question. Mark?
Yeah, so Pete made the same argument that I would make, which is, you know, that it's true that we're better at it for, you know, 100 or 1,000 flies, but when you get to 10,000 and 100,000 flies, you're just not going to do it. Mm-hmm. So you lose the insight that you need from getting enough data. But I was going to just point people, if they're interested, there is a group at Princeton with Mala Murthy that does a lot of work that connects sound from flies with their behavior to try to decode what their courtship songs are. So, you know, it might be fun to have her give a talk as a sort of a companion to see how you integrate sound along with movement.
Hey, I think that's it. Just join me to thanks, Kristin.
Thank you for having me.
Thank you so much.
Thank you, Kristin, and thank you everybody for coming.
Thank you.
Thanks, Kristin.
Thank you. I'll see you in a little while.
That was really nice, Isaac. The interaction is really, I've been giving all these recorded talks, like they asked me to record them in advance, and it was just like talking into the void, and it's, yeah.
Yeah, it's really hard to do that.
Hey, Kristin. sorry, I did not get the chance to put myself in onto a schedule. I'm not sure if you still have time slot available today, or maybe we can schedule a brief meeting some other time. I'm really interested in the.
Yeah. Well, let's schedule. If you send me an email, we can schedule a time to talk about whether your data would work with us.
Well, yes. All right. Great. I'll send the email right away. Thank you so much.
Anna, are you there? I think she probably left.
Okay, so are we scheduled to meet at 2? Is that right, Isaac?
Yes. Well, yes, that's 20 minutes.
Okay.
Just leave you some time to rest a little.
Okay, I will see you in a few minutes then.
Okay. Thank you. Bye.